Welcome to this explainer. Today, we are unraveling a biological mystery that has literally puzzled neuroscientists for decades. Be honest, when you hear the word dopamine, what do you immediately think of? It's reward, right? We all know it as the famous pleasure chemical, the molecule that reinforces our daily habits. But, well, the source material we're digging into today reveals a completely different, almost hidden side to dopamine. We are going to see how this chemical is actually doing something so much more complex and dynamic than just, stamping in memories of past rewards.
Okay, let's dive right into this paradox. The fundamental question we're looking at is this: how exactly does one single chemical control learning, movement, and motivation? If you think about it for a second, it's pretty wild. We know dopamine is absolutely crucial for learning because of its role in processing rewards. But we also know that when dopamine-producing neurons die off, the devastating result is Parkinson's disease, which is a disorder primarily characterized by an inability to initiate physical movement. And then, on top of all that, there's motivation, literally how badly you want something. Historically, science has treated these three roles, reinforcement, movement, and motivation, as completely separate jobs. But the fascinating research we're analyzing today set out to see if these three seemingly totally different jobs are actually just different faces of the exact same underlying biological mechanism.
To solve this mystery, the researchers needed a very specific tool. They designed a self-timed movement task for mice. Here's how it works. The mouse hears a brief little cue tone. Then it has to guess the exact right time to lick a spout to get a delicious juice reward. But here is the major catch. The reward window is completely hidden. The mouse only gets the juice if it manages to wait and lick somewhere between 3.3 and 7 seconds after that cue tone. If it gets overeager and licks too early, it just gets an error tone. If it waits too long, a miss tone. Because these mice have to discover this specific timing entirely through trial and error, it provides us with this perfect, real-time window into how they naturally adjust their behavior based on their successes and failures.
And what the researchers found was this totally fascinating behavioral pattern that they actually dubbed the sloshing effect. Imagine you're fine-tuning a guitar string, right? You're twisting the peg back and forth, going a little too high, a little too low, until you hit that perfect pitch. Well, the mice did something very similar. When a mouse licked too early and failed to get its juice, on the very next try, it would slow down and wait just a little bit longer. Now that makes intuitive sense. But what happened when they actually got the reward? Well, that is where things get really interesting.
Here is the massive plot twist of the study. According to classic, old-school reinforcement learning models, if an animal performs an action and gets a reward, that exact behavior is supposed to be reinforced. The animal should want to repeat the exact same winning action. So in theory, if the mouse licked at exactly 4 seconds and got juice, it should lick at 4 seconds again on the next try. But that is not what happened at all. The huge new discovery here is that getting a reward actually made the mice move earlier on the next try. Instead of repeating the exact same timestamp they just learned, they sped up. Their entire physical vigor was quite literally shifting forward. Now, what's absolutely wild is how this phenomenon maps directly onto the dopamine happening inside the brain. The researchers used these highly sensitive special sensors to measure the exact amount of dopamine released in the mouse's brain right after a trial, and they found that the sheer size of the dopamine spike, which they called the transient amplitude, perfectly predicts exactly how early the mouse will move next. It's almost mathematical. If a trial was rewarded, bam, a huge dopamine spike occurred, and the mouse moved much earlier on the next trial. If it was unrewarded, dopamine dipped, and the mouse moved later. It was this perfectly correlated one-shot update to their behavior, driven entirely by the physical volume of the chemical being released.
Which brings us to an incredibly crucial concept, the propensity to move. This is essentially a measure of behavioral vigor. What this data is clearly telling us is that dopamine is acting way less like some memory maker meticulously writing down the correct answer in a textbook, and much more like a real-time tuning dial for physical vigor. A big, satisfying splash of dopamine essentially turns the dial up, increasing the animal's propensity to move earlier and faster. A dip in dopamine turns the dial down, slowing things back up. It's an immediate, continuous, real-time calibration.
Okay, so let's move to the interrogation phase, because any good scientist is going to try to poke holes in this. You see, in a normal experiment, a thirsty mouse naturally just gets less thirsty as it drinks juice over the course of the session, right? So its dopamine levels might just naturally drop as it gets full, which could coincidentally match its changing movement times. In science, to prove that one thing is actually causing the other, you have to break this kind of simple correlation, or what they call monotonicity. You have to definitively prove the dopamine is actually driving the movement and not just slowly dropping because the mouse is getting full.
So, to prove this tuning dial theory wasn't just some random coincidence, the researchers decided to throw some serious curveballs at these mice. They tested them under extreme environmental changes to map exactly how their dopamine levels would adapt. They completely removed the juice, they rapidly satiated the mice with Gatorade, and they completely changed the timing constraints without any warning. Let's look at exactly what happened.
First up, the no juice test. Astonishingly, these highly trained mice kept right on working, just for the correct beep tone, even when the juice spout was totally disconnected. But without the actual physical juice, their dopamine reward spikes were much smaller and dropped off way earlier in the session. And exactly tracking with that dopamine drop, their timing rapidly shifted later, straight back into the correct window. Then, there was the Gatorade test. The researchers actually paused the experiment and let the mice chug as much Gatorade as they wanted until they were completely stuffed. When they put them back in the task, their dopamine spikes plummeted immediately, and their physical movements drastically slowed down. The motivation was gone, so the chemical tuning dial got turned all the way down.
But what about changing the rules entirely? In what they called the time shift task, the researchers secretly changed the reward window. For example, instead of licking between 3 and 7 seconds, suddenly the mice had to lick between 1 and 2 seconds. The mice had to figure this out completely on the fly. And sure enough, their endogenous dopamine actually recalibrated their behavior. When the mice discovered that new window, their dopamine spikes adapted in real time, pushing their movement timing earlier or later to meet these completely unknown, brand new environmental rules. The dopamine dial was literally actively searching for the new correct timing.
And this ultimately leads us to the final brilliant conclusion of the paper. I actually want to read this quote directly from the source material because it's so good. This entire mechanism is consistent with a stochastic search process that calibrates the frequency of movement. In simpler, everyday terms, it's a giant trial and error guessing game. This is the elegant solution to our central mystery today. Dopamine doesn't have 3 separate distinct jobs. Learning, movement, and motivation are unified into one incredibly simple process. The motivation, how thirsty that mouse is, dictates the size of the dopamine spike. That spike, which is the learning feedback, turns the tuning dial, and that dial directly controls the vigor and timing of the movement. It is all just one beautifully interconnected loop.
So why does all this matter for us? Well, we can actually connect the biology of a thirsty mouse directly to human health. Think about Parkinson's disease. One of the absolute hallmark symptoms is bradykinesia, which is this extreme slowness of movement or difficulty even initiating a movement. If we understand dopamine as a physical tuning dial for the propensity to move, Parkinson's represents a structurally broken dial, one that's turned all the way down. This is exactly why dopamine-modulating therapies, like giving patients dopamine repletion drugs, work as artificial tuning dials. They literally crank that propensity to move back up, so patients can initiate actions again.
As we wrap up this explainer, I want to leave you with a really fascinating question to ponder. We've just seen how a microscopic splash of a chemical can shift the physical timing and vigor of a mouse by mere fractions of a second, all based on whether it succeeded or failed just moments prior. So how is your own dopamine tuning dial subtly adjusting your every move, right now? How is your current motivation, your environment, and your recent successes or failures invisibly calibrating the speed at which you walk, talk, and interact with the world today? It's an incredible unseen force constantly operating in the background of everything we do. Thanks so much for joining me on this deep dive, and keep questioning the science behind your every move.